Over the last few years, liver engineering has become a hot topic around the world due to its potential of generating transplantable liver grafts. However, viable long-term function and the transplantation of engineered organs are still a vision of the future. Recently, in vivo liver lobe perfusion has become a promising strategy to study liver engineering.
A major advantage of this technique is that the in vivo repopulated partial liver scaffold is subjected to physiological blood perfusion. This provides the scaffold with proper temperature, sufficient oxygen, nutrients and growth factors, in contrast to ex vivo perfusion with artificial culture medium. Another advantage is that the remaining liver maintains hepatic function and thereby principally allows long-term survival. However, the in vivo single liver lobe perfusion model is technically challenging and post-operative survival has yet to be achieved. Here we are going to present a novel surgical model with long-term survival as a foundation for further in vivo single liver lobe engineering.
Before we demonstrate the surgical model, we'd like to give a brief introduction to rat liver anatomy. To establish our single liver lobe perfusion model, we selected the left lateral lobe. This is the only liver lobe with a distinct vascular supply and drainage, allowing to create a bypass. This is a 3D reconstruction of the portal venous system in rat. Our focus here is on the left portal vein, which will later serve as a fluid inlet. This is a 3D reconstruction of the hepatic venous system in rat. Here our focus is on the left lateral hepatic vein, which will later serve as